Illumina sequencing is a form of next-generation sequencing technology. It has been used to sequence many genomes and study health and disease. The type of sequencing Illumina uses is sequencing by synthesis, which can produce millions of highly accurate reads of DNA sequences much quicker and cheaper than other methods by the use of a flow cell. There are 4 stages of Illumina sequencing. The first is sample preparation. In sample preparation, DNA is extracted from cells and fragmented into smaller pieces. The purpose of sample preparation is to process the DNA into fragments that are the correct size for sequencing, as well as add adapters or additional short sequences of DNA to the ends of the fragments. These adapters have 3 parts that serve 3 purposes. First is to allow them to bind to oligonucleotides, or short nucleotide sequences, that are tethered to the flow cell surface used for sequencing. Second, adapters also provide a site for a sequencing primer to bind. And third, they allow different DNA samples to be bioinformatically sorted back out later through the use of specific molecular barcode sequences that are different for each sample. The next step in Illumina sequencing is cluster generation. In this step, the DNA fragments are loaded into a flow cell. A flow cell is a hollow glass slide with multiple lanes or channels, each containing a lawn or surface-bound oligos complementary to the fragment adapters. The purpose of the lawns is to bind the DNA fragments to the flow cell. Newer flow cells have a patterned grid-like structure of wells that optimize spacing for the deposition of the DNA fragments, as this picture shows. A DNA polymerase is used to make a copy of each newly bound DNA fragment, using the tethered oligonucleotide as a primer. The original DNA fragments are then washed away, leaving only the newly made tethered copies. Each new DNA fragment is then amplified into a cluster of clones via bridge amplification, as this image demonstrates. In bridge amplification, the adapter at the free end of the tethered new copy of the DNA fragment bends over to bind to an oligonucleotide to which it is complementary, and that oligonucleotide is used as a primer, allowing the tethered copy to itself also be copied. The strands are denatured, and this process is repeated many times, resulting in the creation of clusters, areas of the flow cell containing many tethered copies of the same original DNA fragment. The purpose of the cluster generation step is to quickly make many copies of the same short fragment of DNA in a specific area. Throughout the flow cell, there are many different clusters, each created by making many copies of one specific short DNA fragment. Within a cluster, all of the copied fragments will be the same, but each cluster will be different from all of the other clusters. The next step is sequencing. A sequencing primer and 4 fluorescent nucleotides, adenine, thymine, guanine, and cytosine, are added to the flow cell. Illumina sequencing by synthesis uses a reversible terminator-based method that detects single bases as they are added to the DNA template strand. As a nucleotide is incorporated into each fragment being sequenced, the reaction pauses and the fluorescent signal of each cluster is recorded to detect which nucleotide was added to each fragment. The next nucleotide is then incorporated, and again, the fluorescent signal of each cluster is detected simultaneously. The pause of the reaction is due to a reversible terminator-bound DNTPs, rather than the irreversible DDNTPs, used in Sanger sequencing. The use of reversible chain terminators instead of the irreversible DDNTPs is why Illumina-based sequencing is referred to as sequencing by synthesis, because the replication of the DNA fragment just pauses instead of stopping when a nucleotide is added. The entire fragment being sequenced is actually synthesized in the process of sequencing. The result of the sequencing step is an array of fluorescent tag photos that will be soon rearranged into the correct sequence of DNA. The purpose of this sequencing step is really the purpose of the entire Illumina process, to record the sequence of the target DNA strands. The last step of Illumina sequencing is data analysis. The barcodes that were assigned to each DNA fragment before clustering are now used to sort the separate samples. For each sample, read fragments with similar stretches of nucleotide fluorescent calls are clustered. The fluorescent reads are then aligned to a reference genome. The purpose of this step is to make the qualitative data gathered into a form that can be used for research. The benefits of using Illumina sequencing are large compared to previous technologies. Sequencing by synthesis is a quick, more efficient, and less expensive technology than similar or other sequencing options.